All right, let's jump right into this explainer. We're going to start by exposing a tiny, super common mistake with red blood cell gas transport. It's one of those things that literally, instantly costs you crucial exam marks. You know the feeling? You walk into the exam, you see a question on gas exchange, and you write down this perfectly factual textbook description of oxygen moving through the blood. You feel great about it. But then the results come back and no way, you've completely missed out on the top band. So why does that happen? Well, it's usually because you relied on vague language, like saying oxygen just moves into blood, instead of explicitly stating that it diffuses and binds to the heme group of hemoglobin to form oxyhemoglobin. You see, the examiners aren't just reading casually, they're actively hunting for those precise definitions. And you really, really need to pay attention to this because this specific topic, section 8.2, the role of red blood cells in oxygen and carbon dioxide transport, accounts for an enormous 15% of your total marks in paper 2. That is huge. That's a massive chunk of your final grade sitting in just one single syllabus area. We're not just talking about minor point deductions here. We're talking about full grade boundary shifts, entirely depending on how you handled this exact content.
And here is the harsh reality, coming straight from the Cambridge Principal Examiner reports. Ordinary revision just flat out fails when you use fuzzy language. The examiners point out time and time again that candidates consistently confuse key terminology. For example, mixing up hemoglobin's affinity for oxygen with its percentage saturation. They are not the same thing. You absolutely must define your terms clearly before you start throwing them around in your extended responses. Don't just cross your fingers and hope the examiner guesses what you mean, because they definitely won't.
So, the real question we need to ask is, why do so many incredibly well-prepared candidates just completely freeze and lose marks when they see a brand new experimental context on the page? Well, the syllabus constantly emphasizes that top-performing students show a connected, conceptual understanding that they can easily transfer to unfamiliar contexts. If you're just memorizing facts in a vacuum, well, the second they throw a novel stimulus at you, like, I don't know, a weird new graph on llama hemoglobin at high altitudes, it's going to completely throw you off balance.
But don't worry, the secret to overcoming this freeze is something we call the method mark strategy. This is where we focus entirely on the how, rather than just reciting biological facts. Cambridge rewards process. They want to see exactly how you handle information. When you adopt this mindset, you prove to the examiner that your brain does way more than just simple rote memorization. You're actually proving that you can think like a real biologist under pressure.
Just look at the stark difference between a flawed, generic student answer and an actual top-band response. A generic answer just states, the oxygen moves. Okay, sure, it's descriptive, but it's totally empty. A top-band response, on the other hand, locks in maximum points by clearly linking the high partial pressure of oxygen in the lungs directly to hemoglobin's increased affinity. That's a real game changer. That is exactly what examiners mean when they say they want to see your theoretical architecture at work, connecting a cause directly to its biological effect.
Okay, let's really internalize these top examiner warnings so you never fall into these specific grade-destroying traps. First up are those vague answers we just talked about. Second, forgetting to check your units, which is just a completely unforced error, right? Third is misusing or confusing those key terms. And fourth, entirely ignoring the specific stimulus context that's provided in the question. If the exam gives you a highly specific scenario, your biological reasoning absolutely has to apply directly to that exact scenario. No generic answers allowed.
Now, in just a moment, I'm going to reveal the ultimate examiner clue that changes the answer completely. It's a single word that can literally shift your grade. But hold on. First, you need the framework that actually makes that clue completely obvious to spot. If you try to jump straight to the answer without this structure, you're going to fall right back into that descriptive trap. So let's look at how we actually build that structure.
This is where your theoretical architecture comes in. It's absolutely crucial. Cambridge heavily rewards students who show a connected, conceptual mastery. You really need to view section 8.2 not just as an isolated chapter, but as a giant, interconnected network. Ask yourself, how does gas transport connect to the cell surface membrane? How does the formation of carbonic acid trigger oxygen unloading in respiring tissues? It's those biological cross-connections that push you into the highest scoring bands, because they prove you actually understand the big picture.
So, whenever you encounter an intimidating problem, let's say it's a super complex data table on the Bohr shift, I want you to rely on this exact 5-step procedural framework to systematically process it. Step 1, deconstruct the question. Step 2, extract the relevant partial pressure data right out of the stimulus. Step 3, select your analytical framework. Step 4, execute the analysis with incredibly clear notation. And Step 5, present it all using that precise terminology we've been emphasizing. It works every single time.
You can think of your data interpretation skills as the bridge between the biological theory of gas transport and the actual marks awarded on the page. Knowing how hemoglobin works is great, sure, but extracting the specific partial pressures from a really complex dissociation curve, or interpreting the causal relationship of the Bohr effect from that graph and then drawing an evidence-based conclusion, that is what actually gets you the points. And to correctly execute Step 3 of our framework, you absolutely have to match your statistical analysis tool to the specific data type. For example, comparing mean red blood cell volumes, you must use the t-test. Got categorical data, like blood type frequencies? That's chi-squared. And for looking at relationships between variables, like carbon dioxide concentration versus blood pH, you're going to apply correlation tests. Seriously, using the wrong test on the right data will cost you everything in this section, so be extremely careful. Remember, examiners don't just want to see a magic final number appearing out of thin air. By strictly following a standardized processing sequence, which means identifying key elements, applying relevant biological principles, showing incredibly clear working, and finally verifying your results, you're mapping out your scientific reasoning step by step. They want to see the transparent, logical progression of exactly how you arrived at your conclusion. Now, reaching the highest cognitive domains requires you to critically analyze and evaluate evidence quality. And you do this by aggressively identifying the limitations and assumptions made in the stimulus material. When you're looking at a practical investigation involving carbon dioxide transport, be aggressively curious. Ask yourself, what assumptions did the researchers make about the temperature of the blood samples? What are the limitations of their measuring equipment? Could there actually be an alternative interpretation of this data? All of this, is exactly what examiners mean when they talk about higher-order assessment objectives. They want to see you actually weigh the evidence, not just describe it. When you evaluate alternatives and compare different approaches, you are handling the information exactly as a professional scientist would. And most importantly, notice that you always have to justify your conclusions using the actual stimulus data provided to you in the exam. So, the absolutely crucial takeaway here is elevating your response from merely descriptive to highly analytical and evaluative. Stating facts shows foundational knowledge, which is fine, but it stops there. The evaluative level, where you actively compare approaches and justify your conclusions using those identified limitations, is what truly propels your answer into the absolute top tier of the mark scheme. And please never forget that true comparative analysis means highlighting both similarities and differences. It's actually incredibly common for students to aggressively list every single difference they can possibly find, and then completely forget to state the blindingly obvious similarities. Every comparison has to be supported with evidence to count, and you need to use clear comparative connectives to tie the whole thing together. Okay, so we've mastered the theory, we've got the 5-step data frameworks down, and we know the critical evaluation requirements. You essentially have everything you need to crush Section 8.2. But what happens to your marks if you misinterpret the command word at the very beginning? Honestly, none of this hard work matters. Literally, none of it, if you misread the hidden instruction dictating the entire mark scheme right at the start of the question. And that brings us to the hidden cause of lost marks, command word confusion. This is the single examiner clue that changes everything. It's the silent killer of top grades because it completely dictates whether you should just describe hemoglobin or actually evaluate its efficiency. So what exactly do these different command words demand of you? Subscribe to our YouTube channel Ace the Exams at acetheexams_IGCS to excel in your Cambridge examinations. Join us in the next explainer where we'll diagnose exactly how these command words silently control your final grade. Until then, how are you going to change your approach to the very next exam question you see?